I'm going to talk about our project in developing effective vaccines against C. difficile infection. So here shows my presentation outline. First, I'm going to give you a brief introduction to C. difficile infection and the current status of vaccine development against infection. Then I'm going to move on to specifically talk about our project in developing recombinant protein-based vaccines against C. difficile colonization. So a brief introduction to the pathogen. C. difficile is a gram-positive toxin-producing, small-forming anaerobic pathogen. It's a leading cause of nosocomial antibiotic-associated diarrhea in the developed countries. Moreover, there is a continual rise in the severe infection worldwide, which is partially caused by emergence of more virulent strains like Rab-type 27 strain. So these more produce more toxin, they have a higher sporulation capability, they develop more antibiotic resistance. In addition, they produce additional strain called CDT. It's a two-unit toxin system. So currently, top 5 Rab type C. difficile circulating in the United States includes Rab type 106, Rab type 14, Rab type 2, Rab type 20, and Rab type 27. So more than 90% of infection cases had healthcare exposures, including both inpatient and outpatient. So unfortunately, elderly people, especially for people over 65 years old, have highest infection rates. So currently, C. difficile is listed by CDC as one of the top 3 urgent antibody resistance threats in the United States. So the infection has surpassed Staphylococcus aureus as #1 hospital-acquired infections in the U.S. According to CDC report in 2017, there was almost 220, more than 220,000 infections with around more than 12,000 deaths. So it's a severe problem in the United States. So the symptoms of C. difficile infection range from the mild diarrhea to intestinal inflammation to severe cases of fatal pseudomembrane colitis. So the symptoms are mainly caused by 2 large protein toxins, the toxin A and toxin B. So here show classic risk factors for developing C. difficile infection. These risk factors include antibiotic therapy for different other conditions, advanced age, especially for people older 65 years old, as well prolonged stay in healthcare facilities. Among these risk factors, antibiotic therapy is the single most significant risk factor for developing C. difficile infection. In addition to these classic risk factors, other factors can also significantly increase the opportunity developing C. difficile infection. These risk factors include different inflammatory bowel disease situations, gastrointestinal surgeries, and the use of gastric acid suppression agents such as PPIs and different immunosuppression situations like cancer patients. So many cancer patients will develop a C. difficile infection during the cancer treatment because cancer patients usually undergo chemotherapy, which on one hand can compromise the immune system of the patients. On the other hand, can disrupt the intestinal microbiota, which actually is the major risk factor for developing C. difficile infection. So currently, standard treatment for the infection is to use very few antibiotics, okay, including vancomycin, metronidazole, and fidaxomicin. Because of the emergence of antibiotic resistant strains to these antibiotics, especially the strain resistant to metronidazole. So this drug was no longer as recommended as the first line of treatment in the United States. So there are very few options left for treating C. difficile infection. So in addition, the treatment of antibiotics will lead to very high rate of recurrence because of the treatment, okay, will further disrupt the intestinal microbiota, which is the major risk factor for developing infection. So the major virulent factors of C. difficile are two large protein toxins called toxin A and toxin B. They are both glucosyl-transferases. And the major known intracellular targets of ROTTPases, these toxins are ROTTPases, okay, including ROA, RAC1, CDC42, which are the major intracellular molecules regulating a spectrum of cellular activities. So both toxins, okay, share very similar domain structures, including the N-terminal glucosyltransferase activity domain, followed by is the cysteine protease domain, CPD, and the central pore formation and translocation domain, and the C-terminal receptor binding domain, which can bind to receptors on the host cells. So in addition to these large protein toxins, approximately 17 to 23% of C. difficile isolates produce a third toxin called the binding toxin. So it's a two-component toxin system. One component codes for the enzymatic activities. Another unit codes for a binding function, which can bind receptors of the host cells. So currently, no vaccines against C. difficile infection has been licensed. However, C. difficile vaccination could be a most effective approach over a range of C. difficile risks, especially when being used post C. difficile infection treatment to prevent recurrences. So because the major virulent factors of C. difficile are toxins, therefore, efforts in developing vaccines have been put on focusing on targeting toxins, especially two large toxins, toxin A and toxin B. So, and this paper actually summarizes the current C. difficile vaccine candidates in clinical trials. These vaccine candidates mainly target the toxins, especially toxin A and toxin B. The first one is from the product is from Pfizer, and this vaccine candidate actually includes genetically and chemically inactivated toxin A and toxin B. It's on the phase 3 clinical trial. And the recent update from the phase clinical trial showed that the product didn't meet the goals, okay, and it's not promising. So the second vaccine candidate is from the Valneva, the company. So this vaccine candidate is a fusion protein containing the receptor binding domains of toxin A and toxin B. It was on the clinical phase 3 trial, but so far there is no update on this vaccine candidate. So the third one is from Shire. It's this vaccine candidate is orally delivered non-toxigenic C. difficile spores. And because the major virulent factors are of C. difficile toxins, in fact, if the strain don't produce toxin, they will not cause any disease symptom. Therefore, the non-toxigenic strains can be developed as potential vaccine candidates or as a delivery vehicle to carry other antigens against C. difficile infection. So this one is also on the clinical phase 3 trial, and so the fourth one is it's an antigen from GSK. So very limited information is available for this vaccine candidate, but the information so far I got is still kind of a toxoid, okay, in activated toxin A, toxin B. And it was on phase 1 clinical trial, but so far no information available on this product. The fifth one is from, it's called C. defense from Sanofi. And these vaccine candidates include chemically inactivated toxin A, toxin B by formalin. And unfortunately, this after clinical trial 3, this product was announced non-successful. Okay, it's terminated and didn't meet the goals. So over the years, my lab also has developed recombinant protein-based vaccine targeting the both toxin A and toxin B. And here is illustration showing the domain structures of both toxin A and toxin B. So one of the construct we generated, the vaccine candidate called TCD169, so which includes immunodomains fragments of both toxin A and toxin B. So in this fusion protein, and we fuse the GT glucose transport domain, the cysteine protein domain, CPD, and the receptor binding domain with the receptor binding domain of toxin A to make the vaccine candidate safe and non-toxic. We generated introduced pod mutations, okay, in the catalytic domains, which actually remove the toxicity of the enzymatic active domains. So based on this, we went further. We fused the TCD169 to some line of flagellin C, which is a known potent adjuvant protein, okay, which can significantly enhance antigen immune responses by stimulating toll-like receptor 5. And we have published and reported and published these two recombinant protein vaccine candidates in recent publications. In animal models, these two vaccines showed very good protection against C. diff infection in vivo. So, however, as I mentioned, and the major challenge of a C. diff infection is the recurrence, right? Therefore, the recurrence actually is mainly caused by the persistence of a C. diff cell in the gut. So therefore, the vaccine should not only target toxins but also target C. diff colonization with a goal to prevent both the disease symptoms and reduce the recurrence and the transmission. So because actually over the years and many surface exposed proteins and molecules has been explored as potential vaccine candidates or vaccine components, right? And which can induce protective immune responses against C. difficile colonization and adhesion. And his list of surface molecules, including proteins and polysaccharides, has been explored. And here is an illustration showing the vaccine candidates, including the toxin-based vaccine candidates, as well as the surface proteins or polysaccharide, and on the vegetative cell surface, right, which can be explored as a potential vaccine candidate. And also, effort has been put on developing vaccines against spores, okay? And because the spores are very stable, it's hard to remove, okay? Therefore, targeting a C. diff spore is a rational design against C. difficile colonization, especially the spore colonization. So in the lab, we try to develop vaccines targeting the C. difficile colonization. So we reasoned a recombinant fusion protein containing the C. difficile flagellin FliC, protein FliC, and the Kappa Kappa protein FliHD would be a effective vaccine candidate against C. difficile infection. So and we found both actually FliC and FliD proteins have been implicated in the attachment of C. difficile cells to a mucosal layer of the intestine. And previously, we have showed that FliC immunization alone, okay, can provide it significant, even though partial protection against infection in mice and hamster model infection. And in addition, FliC can regulate the toxin production and also the FliC gene transcription. Okay, therefore, we think a recombinant fusion protein containing these a protein would be a good vaccine candidate. So then we did a little bit bioinformatic analysis. We mined the syphilis-D, from different toxinal types and different rab type acidificil strains. Okay, toxinal type including strength producing all three toxins, or produce only toxin A, toxin B, or only produce the toxin B, or don't produce any toxin. Okay, so we found there is no much variations among the same rab type of C. cell. Then we did compare a detailed line by line homology comparison between the in CDVCO FliC and FliD among different toxin type strains and the rab type strains. And as you can see, the FliC and FliC are pretty much conserved. Okay, the blue regions are the conserved sequences. Only the white regions are different between the different strains. So then we generated a fusion protein with clonal expressed in the gram-positive expression system, which is a Bacillus mandatorum. So Bacillus mandatorum is a soil microorganism, has been used in industry for producing different proteins, right? Different applications. So it's safe, and in addition, Bacillus is a gram-positive expression system. It doesn't have the LPS, lipopolysaccharide issue. So then we after generated the fusion protein, we immunize the mice. Okay, then we collected the serum and fecal samples of an immunized group, and we found immunization of FliC can induce significant anti-FliC responses, antibodies in serum, IgG antibodies and Ig antibodies in serum, as well as the IgG antibodies and Ig antibodies in fecal samples. Okay, and then we further challenged the immunized animals. Okay, as well the control groups, which immunized with PBS. The immunization was carried out by intramuscular approach routes in the presence of alum as adjuvant. So, and here shows challenging results. The first one shows a survival curve of the animals immunized with FliC actually with two different dosages, as well as the control group immunized with PBS. And there is significant reduction in survival in the immunized mice. Okay, and also we monitor the weight changes in three group of mice, similar to humans, right? Mice also develop weight losses after developing C. difficile infection. And also we found a significant reduction in weight losses in the immunized group. And also we monitored the diarrhea symptoms, okay, and we found that there is significant reduction in the diarrhea percentage among immunized group. Then we also determined the toxin levels and the spore numbers, okay, the C. difficile spore numbers, and which used to challenge the mice, okay. and in the fecal samples of the mice, right, control group mice and the immunized group mice challenged with C. difficile R291, which is the epidemic strain, we found that there is a significant reduction in toxin A and toxin B, as well as the spore numbers used for in challenge, okay, in fecal samples. Then we asked, okay, how the immunization provide protection against infection against a hypervirulent strain R291. Okay, so then we collected the anti-fleas serum from immunized mice. Okay, then injected, okay, the anti-fleas serum to the naive mice. Okay, our injection and we challenge the different group of mice, okay, we including the control, okay, with the C. difficile strain R291 spores. And then we monitor the survival, the weight changes, as well as the percentage diarrhea. So we found actually anti-fleas immune serum provides mice significant protection against infection with C. difficile strain R291. It shows survival curve, right? Here's the immunized the mice challenged injected with anti-flea serum. Here's mice actually injected with the pre-immune serum as a control, as well injected with PBS as an absolute control, right? And so there is a significant protection against C. infection. So we also determined the toxin levels, as well the C. difficile numbers in fecal samples of mice, okay, administered with anti-fleas serum, as well a PBS control, and followed by challenge with C. difficile R291. And we found there is significant reduction, okay, in toxin A, toxin B, as well R291 spore number in fecal samples. So then we determined whether the anti-fleas serum can inhibit adhesion of C. difficile cells to HCD cells. Okay, HCD is a human intestine cell line, and we found yes, anti-fleas serum can significantly inhibit adhesion of CD cells to HTC cells, and we diluted, okay, the anti-fleas serum, and then we add the diluted serum to the mixture of the C. difficile cells and HT cells. Okay, then we determined the adhesion percentage, and we found there is significant reduction. So a brief summary about our work. So we found immunization of mice with a FLICD fusion protein-induced potent IgG and IgA antibody responses against FLICD, protected mice against infection, and decreased toxin levels and spore levels in feces after infection. And we've also found anti-FLICD serum could inhibit the binding of C. difficile vegetative cells to the human cells. And these results, our results suggest FLICD may represent an effective vaccine component against C. difficile infection, which can be used in combination with a toxin-based vaccine candidate. So finally, I would like to thank the major players in this project, especially Shaohui Wang, who is a senior person in our research team, and also other contributors in this project. And we thank NIH as well Anthony Foundation for supporting our project. Thank you so much, and I'll be happy to take any questions. Thank you.